Okay, so let's talk about our pathogens today. So remember, we talked about our immune system. We talked about all the things that our immune system can do. I'm going to turn off this front light. We talked about all the things our immune system can do. We have B cells, we have T cells, we have all these antibodies that protect us. But we still get sick. And you might be asking yourself, well, why? And it turns out because no matter how many defense systems we have, pathogens have ways to evade them. So remember, pathogens are going to show pathogenicity. What that means is the ability to cause disease. And the virulence of an organism is how easily it can cause illness. So if an organism has a high virulence, that means that it can cause disease easily. If I have a low virulence, it's harder for me to cause disease. Now, to cause a disease then, a pathogen has to do a couple of things. It first of all has to be able to get into our bodies. So it has to be able to evade our defense systems. So to do that then, that means that it has to be able to gain or access entry, maybe through a cut in the skin, maybe through ingestion, but it has to figure out a way to get through our defenses. Once it's inside, it has to quickly figure out how to attach to our tissues. If it's circulating, it knows it's going to run into a white blood cell. So it needs to figure out how it can cause an infection and where it can attach. Once it's present, it has to evade our immune system. That's going to be tricky. And then as it starts to grow, a lot of times they'll result in some sort of damage. They might be like our streptococcus that we talked about with strep throat, where they're lysing our blood cells, which is why it's so painful to have them. They might be like Vibrio, where they're digging into our GI tract. So they're going to cause some sort of damage just by being present in the body. Now there's a couple of ways that pathogens can gain access to us, but their preferred portal of entry is going to be the respiratory tract. That's the most common way, by inhalation or having us breathe them in. But they can get in a bunch of different ways through any of our mucous membranes. So remember, we have mucous membranes present in the respiratory tract, but we also have mucus present in our GI tract to help move product along, our genitourinary system, and then also in the conjunctiva of the eyes. So these are all going to be areas where our organisms can be able to figure out ways to cause infections and to embed themselves in our bodies. So let's go through some of these and see some of the ways that pathogens can get in. The respiratory tract. So as we mentioned, this is usually the easiest way for a pathogen to get in. They can either be inhaled through being close to somebody who is sneezing or coughing. This is usually the most common types of infections that we see. And so a lot of the ones that we're going to see with this are going to be ones like the common cold. They're going to be ones like the flu, tuberculosis, whooping cough, pneumonia, strep is spread this way, measles, diphtheria. So all of the big ones that we're used to seeing and the ones that are the most easily contracted are going to be ones that are going to separate and move through the respiratory tract. This is why masks have been so important with COVID. One of the ways it's spread is through respiratory. That's why they recommend masks. But it also, honestly, the masks, I know we don't like them, but they actually help us with things like colds and the flu as well. So our flu numbers and cold numbers are down because we have our masks on. So when we're out in public, we have less of a chance of contracting these droplets. The GI tract is going to be the next one. And this is where we would see things like cholera that we talked about before, but also salmonella, like we talked about with typhoid Mary, Shigella. This is also how we can get exposed to our friend Helicobacter, which is the one that causes ulcers, and then things like CBOT. So when we eat food that has a high level of bacteria present in it, or a high level of viruses present in them, that food will move through our GI tract. And unfortunately, if it can survive the stomach acid, it can move into the upper and lower GI and cause infections. We call this a lot of times the fecal oral route. And what that means is that if you have somebody who's sick, let's say we had typhoid Mary, remember typhoid Mary? She didn't show signs or symptoms, but she was spreading Salmonella typhi in her fecal matter. Now she was washing her hands, but what did we learn about normal flora? It sticks, no matter how carefully you wash. And part of her normal flora had become Salmonella typhi, unfortunately. So let's say that we have somebody wash their hands, or we have somebody use the bathroom and not wash their hands. Now they have fecal deposits on their hands. And it could, very small. They don't see them. It's just microscopic. Now they go and prepare food, and as they prepare food, that microscopic organism transfers to the food. So that would be the fecal origin. And then as you eat the food then is contaminated with these organisms, and that's how you get ingested and contaminated inside the body. So some of the outbreaks that we often see with things like vegetables or fruits, we have a lot of recalls for those. A lot of times veggies, remember, are grown in fertilizers. What's in fertilizer? Manure, which is fecal matter. So that's how we get exposed to a lot of things through our veggies. Also, if people are harvesting veggies, if they don't have clean hands, then you can get things that way. So that's one of the reasons why we see a lot of recalls with vegetables and fruits. The genitourinary system, this would be more of our STIs or STDs, however you want to refer to them. So sexually transmitted diseases or sexually transmitted infections. So things like gonorrhea, syphilis, chlamydia, herpes, all of these are going to be spread usually through direct contact. So having some sort of direct contact sexually will be able to introduce these viruses or these bacteria. The conjunctiva is the part of the eye that's a mucous membrane that helps to protect the eye. It helps to make sure that our eye stays safe and moist with secretions. Unfortunately, there are a few organisms out there that can cause chronic inflammation of the conjunctiva. And an example of this would be something like chlamydia. We think of chlamydia as an STI, but different species can cause issues in different locations. So chlamydia trachomatis is one that can cause an eye infection that's persistent. The problem with this eye infection is it causes swelling of the membrane tissue. And it causes the eyelashes to actually start to curve in because of the swelling. So your eyelashes are normally like this. The swelling of the eyelid causes them to curve down. So now when you're blinking, you're dragging your eyelashes across the surface of your eyes. This can result in scarring. And if it's a chronic infection, it can actually result in permanent scarring. So you see here, this is a patient who has permanent scarring present because they keep getting reinfected with this eye infection and they're not able to treat it. Maybe they don't have access, maybe they're not able to get to the right antibiotics. So now what's happened is where their eyelashes have scraped across their eye, they've had scarring and inflammation, and it creates almost like a cataract. So they now have film over their eye that cannot be removed. And so they've permanently lost sight in that eye because of this chlamydia infection. The second easiest route for organisms is to be able to try to find a breach in our skin. So remember, our skin is our largest organ, it protects us, but if there's any kind of break in our barrier, the microbes can get in. Usually where they try to infect is along the hair line, along the hair molecules, or also along the sweat glands. So they'll try to sneak in if they can through where the hair is coming out of the skin. That's like kind of a weakening in the system. They'll try to sneak in where sweat glands are. But for the most part, if your skin is intact and dry, you're fine. It's when we have moist skin or when we have skin that has cuts or openings that we run into situations where the bacteria can get inside. The third portal of entry would be the parenteral. And so this would be where you're having a deep cut into the skin. So like a puncture, a bite, a wound, maybe surgery. Something where you split the skin, but it's deep. And so now they can get into regions that are normally anaerobic. They can get into regions that will cause a faster infection and allow them to spread quickly. So this is one of the reasons why we actually so careful when we have surgeries. We are opening your body up. We're exposing it to anything in that room. So we have to make sure we're as sterile as possible. Here's an example of a MRSA infection. This is a bite infection. So anytime you get a deep puncture, you're prone to having bacteria or viruses move in. Now, every single organism out there is going to have what we call a preferred portal of entry. And what this means is that they're going to have to go through a certain route to cause an infection. So we might get exposed to a pathogen, but if it's not through its preferred route, it's not going to be able to cause illness. So, for example, smallpox. Smallpox was spread through touch. We called that variolation. And what that meant was the pustules would break open. If you picture something like chicken pox or being covered in bite or like mosquito bites, that's kind of what they look like, little bumps. They would break open and they would ooze. And if I touch somebody, then that would spread the virus. Another example would be something like Streptococcus pneumoniae that is spread through droplets and then inhalation. Now, here's where I mean about the preferred portal. If I was to lick smallpox, I don't know why I would, but if somebody had smallpox on their arms and I licked the pustule, I would not get sick that way. It can't cause illness through the stomach acid. So its preferred portal is touch. If I digest it, it doesn't work. The same is true with strep. Strep pneumonia prefers to be inhaled. So if I have a cut in the skin and I get strep on it, it's not going to cause an infection. That's not its preferred entry. Does that make sense? So each organism out there has a certain pathway it follows. If it's on the wrong path, it can't cause illness. Okay? Salmonella typhi. It can cause illness if ingested. But if I was like to touch it, like let's say typhoid Mary and I shook hands, I would not get ill from that. So just through touch, it wouldn't be able to be spread that way. So every organism is going to have that preferred pathway that they're going to use. Now, I mentioned before that some organisms can cause illness easier than others. And the way we can determine how easily an organism causes illness is by looking at its ID50 and LD50 levels. ID50 is the infectious dosage. And what this means is if I was to set up a test, this is the number of cells that each organism needs to have and expose to a host to cause illness in 50% of my test subjects. So if I have 100 rodents in front of me and I expose them to a certain number of cells, I keep exposing them, keep increasing the number of cells until I see what level causes infection in 50% of my rats that I'm testing or my rodents I'm testing. Does that make sense? So that's my ID50. We then take that data and we say, okay, in rats, it caused around, for Vibrio, around 100 to 10,000 cells to cause an infection in 50% of them. Some are very low. Shigella only requires 10 cells. That's like phenomenally low. Most organisms are going to average in the thousands, but some, like I said, they're very, very capable of causing disease. They have a high virulence. So that's my infectious dose of 50. Now, an infectious dose is different than an LD50, which is a lethal dose. This is the number of cells that I need to introduce to actually cause death in an organism. So now I take my rat population and I expose them to an increasing number, keep increasing until I see 50% of them pass away. And then that's going to be my lethal dose. Now note, I have different levels for inhalation and infectious doses versus lethal doses. So for example, here I have anthrax. Anthrax, when it's inhaled, can have an LD50 of about 50,000 endospores. So what that means is this is an organism that prefers to go through the inhalation route. It prefers to cause infections in the lungs. It can also, though, go through the oral route. It can also be ingested, but I have to have a much higher dosage, 250 to over 1 million endospores. So what this means then is, remember when anthrax was out there back in the early 2000s, it was being used as a weapon. A lot of people were worried about it because they were saying, we don't need a whole lot to cause an infection. That was true to a point, but it really depended on how it was going to be administered. If it was going to be inhaled, then yeah, that's a much lower number than if they tried to poison like our food supply or our water supply. Does that make sense? So we have to look at how the pathway is occurring, and that will dictate what our ID50 is or our LD50, because they can be different for different organisms. So if I'm a pathogen and I'm trying to cause an illness, the first thing I have to do is I have to be able to cling to the surface of a host. so that I can't get removed. So for example, if I'm trying to cause illness on the skin, I don't want to be easily removed and get pushed away before I'm even able to get inside the body. Once I'm inside the body, the next trick I have to do is I have to try to avoid phagocytosis. I need to get away from those white blood cells that are circulating and trying to destroy me. Some organisms do this with producing capsules. Some are going to do this by just trying to get into a host cell as soon as possible, but they're trying to evade defenses. They're also going to try to do penetration. They're going to try to get into a tissue or into a cell as soon as possible so that they can try to evade our innate immune system. When they're inside of us, that means that now we have to go to another level. We have to go to T cells. We have to go to our third stages of immunity. And so they're going to try to get inside as quickly as possible. Once they're present, they're going to start producing their own enzymes and toxins. And this is usually what will make us ill. It's their waste products that they're producing. So, for example, with cholera, we mentioned that the toxins that they release occur when cholera is lysogenic. What does that mean again? What does lysogenic mean? Excellent. When cholera has a virus in it, the toxin level that it produces is higher than normal. And so this is why with cholera, if it has a virus in it, if it's lysogenic, it causes an illness. It produces a high level of toxin in our bodies. The problem with toxins is our bodies react to them quickly. It's like a poison being released. But the toxins also don't stay just locally. So with cholera, it doesn't just stay in the GI. It can travel in the bloodstream and it can cause other issues in the body. So that's the problem that we run into with toxins, is they're not just isolated. They can start circulating and spreading with our circulation system. Now, with their adherence, one of the ways that they can adhere to our cells is through the use of things like the glycocalyx. Remember, we talked about glycocalyxes. Those were those sugar or protein coats. Think of things like plaque, to build up these glycocalyxes. They can also try to stick through the release of like Fimbriae. We talked about how E. coli had these little fringes on them. They can use those like little hands to stick. TB, Mycobacterium tuberculosis, it has those mycolic acids, remember, in its cell wall that are fatty acids that make it harder for our cells to attack it. Some organisms like strep, they have special proteins that knock out our complement system. Remember, we talked about complement as being like a sequence of dominoes that triggers an immune response. Well, strep produces a protein that basically stops the domino before it can fall. And so it stops complement from occurring. We also can have certain ways that they can attach with things like tapered ends that are like hooks. So here's an example of a parasitic worm that has this hook to it. Treponema pallidum, bless you, which is syphilis. It's corkscrew shape. At the end of each of its corkscrews, it has kind of a hook to it, which allows it to dig or embed into the tissues. So it's hard to get rid of them when they've embedded. So what this means then is that the bacteria have a lot of tricks up their sleeves to stick or to adhere. So it's harder for us to get rid of them in our bodies. I mentioned before they can also produce capsules. Here's a bunch of organisms that do produce capsules in the body. Remember, capsules make them bigger, so it's harder for our bodies to destroy them. The white blood cells will approach, but they can't figure out how to phagocytize them because they're too big. I told you an analogy before when we talked about capsules. It's kind of like when you get a huge hamburger, how you have to like cut it, because it's so big you can't figure out how to bite it. So some of the organisms that we see that a lot of times will have capsules or some sort of protection to them would be things like Streptococcus pneumoniae, Klebsiella pneumoniae, Haemophilus influenza. Now, careful with that. Haemophilus influenza is a bacterium. It's not the flu virus. It's actually a bacterium that causes respiratory infections. Bacillus anthracis, Streptococcus mutans, and Yersinia pestis. What did Yersinia pestis cause again? Anybody remember?
Black plague.
Good. Black plague. Good. Excellent. Now, there are enzymes that they can release that help stop our immune systems that contribute to their pathogenicity are a bunch of different varieties. And it really depends on what organism we're talking about as to what enzymes they can make. So, for example, Staphylococcus makes an enzyme called coagulase. This causes our blood to coagulate around it. And what that does is it's like kind of creating an igloo. I told you before that staph infections kind of create like a little igloo around themselves. Do you remember that? The way they do that is through coagulates. They coagulate our blood cells around them, and now that coagulated blood is like a barrier. It's like a scab that surrounds around the staph. And the staph is in the middle. And the staph is growing, it's producing toxins, so it's like a little hollowed out igloo in there. And all around it, this hard, dry blood makes it so that our white blood cells can't get through. It's like hitting a brick wall. And this is why if you have a staph infection, they have to pop the boil or puncture it, squeeze the pus out, squeeze the staph out as much as possible. And what that does is it knocks down that blood clot. And now the white blood cells can get back inside. But until that time, they're kind of blocked out. Kinases. These are ones that are also used to digest clots. So certain organisms like staph and strep, if our white blood cells are attacking them and they're starting to group up, what will happen is they'll release kinases that cause our white blood cells to basically be diluted out. So it's harder for our white blood cells to group around them because they're like, it's like, trying to pour water on somebody that's trying to run towards you. It's just like a defense system. Hyaluronases. These are ones that break down connective tissue. These are ones then that are seen with what we refer to as flesh-eating bacteria. You guys heard that term before, flesh-eating bacteria? So flesh-eating bacteria produce this enzyme, and what it does is it destroys the connective tissue that holds your tissues together. And so what this does is it causes the skin to just kind of separate from the bone and muscle and slough off. That's what flesh-eating illness is. I have a very G-rated version of flesh-eating illness coming up because some of the pictures are just kind of horrific and I don't like to show them. Collagenase, these are going to be one that are going to break down collagens. We talked about how collagen can be eaten by some of our organisms. Clostridium and streps are ones that can do that. They can break down collagen in the body. And then cytophores, these are ones that allow bacteria to take iron from a host. So if you have a big infection going on, for example, like if you have a UTI with an E. coli, that infection will cause you to feel tired and it'll cause you to feel run down. And you might have wondered, well, why does a UTI make me feel so tired? It's because the E. coli that's present is taking iron from you. They can't produce iron on their own, so they release these enzymes that pull any iron from your blood towards themselves, and then they use that to grow. So it makes you feel tired because you're not getting enough iron to carry the hemoglobin in your blood. So it's a way that it makes you feel kind of exhausted.
Are they powerful enough to cause liver failure?
Oh, good question. If they get in a high enough number, yes. So they can go septic, they can affect the liver, they can affect the kidneys, and they can cause organ shutdown. So yes. I mentioned the Brazilian model that died from that UTI infection. She had just general E. coli. But the numbers got so high that it affected her.
Did she have like jaundice?
She didn't have any signs right away with the jaundice. Remember, she had it for about 6 weeks and she kept ignoring it. And then when it finally did go septic, she first of all had her kidneys shut down and then the liver shut down. It happened so fast, she never really showed signs. That we usually see where the whites of the eyes turning yellow. She never really showed signs of that. It just all happened really fast.
She had to have had a high pain tolerance.
I think she had a high pain tolerance and then she had just a crazy schedule. So she just kind of kept putting things off. And being a model, I guess, according to the article, is like she had to show up to get paid. So if she missed something, they could replace her and then that would be the end of her career. So in her mind, she just thought, well, it's a UTI, it's not that bad, I'll just keep going. And unfortunately for her, it was a bad gamble. Other enzymes they can use. We talked about our alpha hemolytic strep. We'll look at our plates today. I'll have to pull those out of here in the fridge. We'll look at our strep plates today and we'll look at the hemolytics of them. Remember from our throat swabs the other day? And what we're going to be looking for on our throat swab plates is we're going to be looking to see what kind of hemolysis we have. What we don't want to see is hemolysis like this. This is beta hemolysis. And if we look at our throat plates today, we're going to hold them up to the light and we're going to look to see if we have any clearings. Now, a completely clear or halo around our colony means that the red blood cells are being used as a food source, and that's indicative of a streptococcus, usually strep pyogenes, which is strep germ. A lot of times we'll see alpha hemolysis. That's where some of the blood cells are getting destroyed. Now, it turns out we have a few normal flora that can be present that do very weak alpha. So they use our blood cells as a food source, but their levels are low enough that we never feel sick. So if you see a little green discoloration on your plate, don't panic. Alpha hemolysis does not always indicate a pathogen. We would have to do additional testing to see if it was pathogenic or not. A lot of times we have some normal flora that'll do it. Hopefully what you'll see is nothing on the plate. You'll see a lot of growth, but no hemolysis at all. Glucosidins. So these are going to be enzymes that are going to attack certain types of white blood cells. So, for example, they're going to either attack the white blood cell when it's trying to do phagocytosis, and they could kill the white blood cells that's trying to eat them, or they allow the white blood cell to consume them, and then once they get inside, they release an enzyme that causes the lysosome in the white blood cell to pop too early, and that causes death to the white blood cell. And then the bacteria will try to sneak out before it gets exposed to the enzyme. So they can try to kill the cell as they go along. Now, I mentioned before that we have our flesh-eating bacteria. And again, this is because of this collagenase that they produce that allows them to break down the collagens and then also break down the connective tissue of the cells. So here's a picture of flesh-eating bacteria. And again, this is the G-rated version. And what it's causing is it's just causing the tissue to actually separate from the muscle tissue and the bone. And so if you see this on a patient, you could just basically peel their skin. Just like an onion, you could just peel it. There's not any more of the connective tissue present. Now, one of the things I get asked a lot is what bacteria are flesh eaters? Well, there's several that have a tendency to go that way. So some of the Clostridiums can go that way. Some of the Acinetobacters can go that way. Some of the Streps can go that way. That doesn't mean that all of them do. So for example, I could have a strep that has a genetic variation that allows it to break down our connective tissues. And then I could have a strep that doesn't have it, and then I don't have to worry about having flesh-eating bacteria. So it's not so much the organism, it's what genes they have as to how much damage they can cause. Aaron?
So is it only that enzyme hyaluronidase
Yep.
that causes it?
Yep. The hyaluronidase, and then also if they have a collagenase that breaks down the collagen, it can contribute. The two together is a deadly form. The hyaluronidase by itself is the one that is going to cause some loss or damage to the tissues. So, for example, there was a girl who was ziplining in, Costa Rica. Ziplining in Costa Rica, got her leg caught by one of the zip line ties and got a deep puncture wound or deep wound around her leg. She decided to go wash it off in the river, and when she washed it off, she ended up picking an Acinetobacter up. And unfortunately for her, the Acinetobacter that she was exposed to was one that was able to do flesh eating. She saw that it was getting infected the next day. She decided to ignore it. And the reason she decided to ignore it was because she was flying home in 48 hours. So she thought, well, rather than trying to find a doctor here, I don't know if they're going to speak English, I'll just wait and I'll go home. So when she went home, her leg was, really hurting. She landed. She showed it to her parents. She was around 19. Showed it to her parents. They agreed she should go to the ER. So she went into the ER. They started checking her leg. They went to wash the leg, and her skin started peeling off. Like the skin was just peeling. And they realized that they had a pretty bad infection. So they started working on trying to limit it, isolate it, started putting her in an oxygenic bariatric chamber, trying to see if it was maybe a Clostridium. They were trying everything they could think of. The problem with flesh-eating bacteria is there's not a lot of ways to stop its spread. So if it starts to grow and spread, usually if it has the hyaluronidase enzyme, it also is resistant to a lot of our antibiotics. And so the concern that they started to have was as the bacteria was spreading, they were worried that the longer they tried to treat and just messed with it, the more it was going to spread and it was going to result in more damage. She ended up having to have a leg amputation from the knee down. And that was really the way that we treat a lot of flesh-eating bacteria. If it doesn't respond to treatment within about 24 to 48 hours with antibiotics, they're going to remove that part of the body. And the reason is they'd rather remove it than risk it getting in your bloodstream and spreading throughout. So you have a very short window with flesh-eating bacteria to be able to decide what treatment you're going to do. Now, she's doing great. She ended up getting an artificial limb replacement. She's doing fantastic. And she's fortunate they were able to take it from the knee down rather than waiting and having it go up to her thigh. I mean, it would have just continued to spread. So it's those hard decisions you have to make. How common is flesh-eating bacteria? Do we hear about it in the news pretty often? Yeah, it's not super common. Thankfully, a lot of the bacteria, they don't have these enzymes. They don't have these genes. So for the most part, we're pretty lucky. It's only every now and then that we come across a mutation where they do have it, and unfortunately, it's able to get inside of our bodies. So all of these work together. Oh yeah, Laura.
If she would have went to the doctor earlier, what's the antibiotic have a better chance of?
It would have increased her chances of not having to have so much of her leg removed. So if she'd gone in earlier, the cut was like right around her ankle, they probably still would have had to remove the foot, but it would have just been the foot.
Would have responded to antibiotics better since there's like a
lower number of them. And that's always a possibility, but there's no guarantee. So it's one of those things where you just don't want to wait too long. Again, if you can get in to get treated, the better it is. I've been amazed, when I used to work in the hospital lab, we would have patients come in and would get swabs done of abscesses or whatever, and they'd have medical notes of how long they'd had these abscesses. And it would be like 6 months. And it's like, no, you got to get in earlier, because you're playing a game here. You're playing like a Russian roulette game. You don't know what bacteria you have. So if you have something weird, get it checked out. It doesn't take that long. So all of these are going to work together then to figure out ways to get around our defense system. And again, the whole point for the bacteria is to try to penetrate and be able to grow. And that way we can see that we have bacteria growing in the body and trying to evade our white blood cells that are trying to eat them up. Now, as far as how they can cause damage, our pathogens can damage the host through a couple of different ways. They can do direct damage, so they can cause damage exactly where they're growing. So they can destroy the healthy cells that they've infected. They can multiply and infect other cells and cause other illness. As I also mentioned, though, they can release toxins. And toxins can flow through the bloodstream. They can move into the lymph. And so they can get spread throughout the body. And so toxins a lot of times will show up much farther away than the original source of the infection. A lot of times those toxins can cause hypersensitivity. And again, this is where our immune system overreacts. So I mentioned with a lot of our COVID patients that are on respirators or ventilators, they don't even have a high viral load anymore, but their immune system is overreacting still. Same is true with cholera. We overreact to cholera, which is why we have such a strong immune response with the diarrhea and the upset stomach. It's so quick, but it really is an overreaction. The cholera itself is pretty self-limiting. So toxins then can cause a lot of damage, and they can spread pretty far and wide in the body. Now, the thing about toxins is that then when they spread, they can cause damage throughout. And the toxins can damage in a couple of different ways. They are going to be able to cause death in certain tissues and in certain cells. They can stop the process of complement. They can a lot of times be released as an inactivated toxin that then becomes active when it interacts with our cells in our body. And a lot of times they can also attack antibodies. So toxins themselves can be quite dangerous for us. So we have to be really careful with the toxic level of bacteria. The more bacteria we have, the more toxins we're going to have present. We have to be careful with balancing out those toxins that are in our body. We have two main types of toxins that are seen in the body. We have endotoxins that are going to be formed inside of a cell, and we have exotoxins that are going to be released outside of a cell. Now, different organisms produce different toxins. So we're going to see that some organisms only have endotoxins, while others only have exotoxins. They do not have both. They have one or the other. So they're either going to have an endotoxin or they're going to have an exotoxin. I think this is a good stopping point. I'm looking at the time. So I think I'll stop here and we'll pick up with exotoxins next time, which we start to go into a little bit more detail.